Hey guys, just a quick video tying together some of the photosynthesis and respiration stuff. So we want to keep in mind, right, we have photosynthesis, and some of the things that we use in cellular respiration are products of photosynthesis, and some of the products of respiration are used for photosynthesis. And those things, right, these are organic molecules, meaning carbon compounds, and oxygen, O2 gas. And the things going this way are carbon dioxide and technically water, although water is really exchanged a lot in the abiotic reservoirs that are out in the environment. So we would need to look at photosynthesis as it fits in with these things. So where are these things used and where are these things produced? And we need to be able to do the same thing with cellular respiration. So when we look at photosynthesis, we've got CO2, we know it uses CO2 and we know it uses water. And we know we've got to produce organic molecules, and we're going to end up with some oxygen gas. So we're going to turn this into this. Now, how's that happen? Now, keep in mind that the organic molecules, they're not just matter, right? That's energy locked up in chemical form. And so somewhere in here, we have to add energy. And that's going to come from light, of course, and that's because it's photosynthesis. So we got two parts of photosynthesis. We got the light reactions, the ones that require light. And then we have the Calvin cycle, which does not require light, but it requires all the products from the light reactions. So the light reactions aren't going to use this CO2. That doesn't come into play until the Calvin cycle. That CO2 is going to come in here. But the light reactions use the water. And the thylakoid membranes, which are the innermost membranes of the chloroplasts, we have a lot of proteins lined up, and it's an electron transport chain. That electron transport chain is a little different than the one we talked about in cellular respiration, but not a lot different. The main thing that's different about it is that we start with low energy electrons in water. This is going to be our source of electrons, and we're going to excite those electrons to make high energy electrons. And our high energy electrons are going to be in the form of a molecule called NADPH, which is a lot like NADH. It's basically a molecule that is used to transport high energy electrons. And that's one of the things that's going to go over to the Calvin cycle. We're going to take a protein that's been excited by the light and use it to take electrons from water. And so our water is going to become 2H pluses, 2 protons, plus of a half of an O2. So to make a balanced equation, we're going to need more. We're going to do this more. But don't worry, we're doing this plenty. So we're going to take the electrons from this. I've got 2 positive charges here, no charge on the O2. I've got 2 electrons that I took away. So those 2 electrons are going to get passed from protein to protein. And as they get passed, that causes a shape change in those proteins, and those proteins pump protons across the thylakoid membrane. And so as the proton concentration increases in there, we get a proton gradient. That proton gradient is relieved through a protein called ATP synthase. And so it's a channel coupled with an enzyme. And so it runs these protons through it and uses that concentration gradient, the energy stored in that concentration gradient, it uses that energy to charge ATP. And that's the other thing we're going to produce in the light reactions that are going to be passed to the Calvin cycle. So what we're passing from the light reactions is chemical energy, excited electrons in the NADPH and chemical energy in ATP. But this is energy that once was light and now it's chemical. We're going to pass that to the Calvin cycle, and we're going to transfer that energy into bigger, more complicated molecules, organic molecules. We're going to chain carbons together. We do that with an enzyme called Rubisco, and there's a whole cycle that happens. But Rubisco is the enzyme that it basically adds CO2 and makes it organic. Now, organic molecules are big carbon chains. This is an anabolic process. We need to put energy in, and this is the energy. So we have several places along the way here where we're adding ATP and NADPH to make this happen, to generate these organic molecules. So what you really need to be taken away from photosynthesis is what comes into the light reactions, what goes out of the light reactions, what goes into the Calvin cycle, and what comes out of the Calvin cycle. And then overall, what comes in and what goes out. Right? That's the part that really matters. You should be aware of what Rubisco does. You should also be aware of when I'm pumping those protons, I'm changing the pH, right? Because concentration of hydrogen ions, that's what protons are. That's what pH is. So when we pump the protons across the membrane, we're changing the pH of both of those places. We're raising it where we're pumping from, and we're lowering it where we're pumping to. Remember, lower pH means more acid, means higher concentration of H+. That's really everything you need to know about photosynthesis. Now, when it comes to cellular respiration, remember we're going to start out with C6H12O6, and it doesn't have to be this, but we generally use this because it's kind of the basic model. Everything else can be put into this process. Every other organic molecule can be put into this process somewhere along the way. We need oxygen, but we'll see that we don't need that till later. And we're going to produce CO2, and we're going to produce water. And more importantly, we're going to charge ATP. That's the purpose of this. So cellular respiration is generating ATP for the cell to use. Even if you're a plant, you're not going to use this ATP for normal cell functioning. That ATP is used for the Calvin cycle. You need mitochondria also in your cells. If you're a plant, you have them. Remember, virtually all eukaryotes have mitochondria, including all the plants and all the algae and everything else. So we're going to do glycolysis first. We're going to do pyruvate oxidation, and we're going to do the citric acid cycle. This is sometimes called Krebs cycle. And we're going to do the electron transport chain. So we need to know what goes in and out of each of these things. So glycolysis, this is where that glucose comes in. Glucose comes in here. We're going to split it to 2 3-carbon molecules, and those are called pyruvate. And in that process, we're going to get 2 ATP net. We're going to have to put in 2 and we'll get out 4. So we get 2 ATP and we get 2 NADH. So we also had to add into this 2 ADP plus phosphate plus 2 NAD pluses. And if you remember fermentation, you'll remember that that's about cycling this back so we can have the NAD plus that is necessary to do glycolysis. Right? So this is fermentation. And maybe it's alcoholic and it produces ethanol like in beer and bread, or maybe it's lactic acid fermentation like in us. It's what we do when we don't have oxygen, because oxygen is necessary for the electron transport chain over here, and the electron transport chain is going to make us this NAD+. So we'll cycle it. If we have oxygen, we'll cycle it this way. If we don't, we'll do fermentation. Now we're in the mitochondria. This is just in the cytoplasm of the cell. And as we pass through, we do pyruvate oxidation. So we go from 3 carbons to 2 carbon molecules. So we produce a CO2. We get an NADH out of each one of these 3 carbons. So it should be technically 2 per glucose of each of those. Then we're going to do the citric acid cycle, and we're going to get a bunch of NADH and an FADH for each 2 carbon molecule. Right? So this is going to be acetyl-CoA. That's that 2 carbon molecule. That's going to the citric acid cycle. And a few ATP. I don't remember. It's 2 or 4. It's not very many. So we don't have much ATP at the end of this, but the electron transport chain, and I'm going to jump to the next slide here, where we're going to take all those NADHs and FADH, got a couple of those per glucose, and that oxygen that we had from the beginning. These are the things that are going to go into the electron transport chain. And this is just a string of proteins on the inner membrane of the mitochondria. These are carrying high energy electrons, so they're going to pass high energy electrons to proteins. And just like our electron transport chain in photosynthesis, those proteins are going to get shape changes, because whenever you stick something onto a protein or take something off of a protein, it changes shape. So when you stick the electrons onto it, it changes into a shape that pumps a proton across the inner membrane from the matrix, which is the inside of the mitochondria, to the intermembrane space, which is the space between the outer and inner membrane of the mitochondria. And so it's going to pump protons there. So the pH of the intermembrane space goes down, the pH of the matrix goes up, because we're moving protons, and that's what acid is. The electrons will be passed from protein to lower and lower energy levels. And eventually that final electron acceptor will be this oxygen. And so we're going to use that and some of these hydrogens that we got around, which there are lots of, to make water. So the things we're going to get out are water. We got a big proton gradient. That proton gradient is going to be exploited. It's going to be used again the same way it was in the other electron transport chain. With an ATP synthase, it's going to let protons come back into the matrix of the mitochondria and generate ATP during that time. So we're going to get a bunch of ATP. This is where the bulk of our energy comes from, our chemical energy that's usable by the cell. And we're going to generate some water. So that ATP is really the whole point of this, right? That's the boom. We needed this source of energy in these high energy electrons so that we could do the electron transport chain and get the boom. You may remember we called this NADH the oink oink. So we got the oink oink so we can get the boom. The electron transport chain takes those high energy electrons from these electron carriers and uses it to generate ATP. We talked about how if you can't give the electrons to oxygen, then you can't do the electron transport chain because it requires those proteins changing shape when things are handed to them and passed on. And if they can't pass it to oxygen, that last guy has to hold on to those electrons. The guy before him has to hold on to his electrons. The guy before him. We're not changing shapes. So we're not pumping protons, so we're not generating ATP. Photosynthesis, cellular respiration, all in a nutshell. That's about as tidy as I can make it. Hope it helps. You guys are doing great. Love you.